So my name is John Kelly. I work in the biology department at Loyola University, Chicago. I'm going to be talking to you today about the effects of wastewater treatment on microbial colonization of microplastics. And I want to start out by acknowledging my collaborator on all of these projects, Dr. Tim Holine, also from Loyola University. Whoops. Oh, so how do I do the slide advancing?
Just click once on it with your mouse and then you can use the arrow keys.
Great. Okay, perfect. So microplastics, as we've discussed already, are plastic particles that are less than 5 millimeters in size. And microplastics have been documented as contaminants in marine and freshwater habitats throughout the world. Sources of microplastics to the environment include fragmentation of larger plastic debris that's been discarded to the environment, as well as the accidental release of production pellets used in the production of a variety of plastic materials. However, there are also a number of consumer products that can be sources of microplastics to the environment, including personal care products like soaps and cleansers that can include microplastic abrasives and synthetic textiles. So the use of these consumer products that are potential sources of microplastics can introduce microplastics to domestic wastewater. So the normal use of soaps and cleansers that contain microplastic abrasives will introduce these microplastics to the wastewater. And the laundering of synthetic textiles can also release microplastic fibers into domestic wastewater. So in most urban and suburban areas in the United States, domestic wastewater is treated at a municipal wastewater treatment plant before the water is released to the environment. But because of microplastics' small size, buoyancy, and resistance to decomposition, there's the potential for microplastic to be transported through wastewater treatment systems and be released to the environment. So we wanted to see in an early study done by our group to see if wastewater treatment plants could indeed be potential sources of microplastics to freshwater ecosystems. So, oops. Right. So we did a study on the Chicago River, and we collected samples both upstream and downstream of a large urban wastewater treatment plant that releases effluent to the Chicago River. And we found significantly higher abundance of microplastic downstream of the treatment plant as compared to upstream, as you can see in the figure on the left. The figure on the left is all the microplastics grouped together, but when we looked at individual types of microplastics, including fragments, fibers, pellets, and foam, we can see that all of them were significantly more abundant downstream of the treatment plant. So these data indicated strongly that this treatment plant is indeed a point source of microplastic to the Chicago River. When we looked at these microplastics under the electron microscope, we saw that they were extensively colonized by microbial biofilms. So lots of microorganisms growing on the surface of this plastic. And we looked at this because a prior study by a group from Woods Hole had found microbial colonization of microplastics in the ocean. So we wanted to see what it looked like in this freshwater ecosystem. So the plastics were covered by dense biofilms of microorganisms. So we were interested to find out what types of microorganisms are colonizing these microplastics in a river. So we analyzed the composition of the microbial communities on the microplastic using high throughput sequencing technology. We compared the microbial communities on the microplastic to the surrounding habitats. So the ordination on the right shows you a summary of the microplastic communities from the upstream water in the bottom part of the graph, as compared to the downstream samples, which included downstream water, the microplastic, and ceston, which is basically floating organic material. And what this ordination shows us is that the communities in each of these habitats were distinct in terms of their taxonomic composition. So the types of bacteria that were on the plastic were distinct from the microbes in the water that they were floating in and from the organic material that was also floating in that downstream water. And this was especially noteworthy because we collected the floating organic matter and the plastic using these plankton nets and then collected them by filtering. And so you can see in the image that the floating organic material and the microplastic pieces were intimately mixed together, yet the microbial communities found associated with the organic material and the plastic was distinct. When we looked at these communities in more detail, so this stacked bar graph shows you the 20 most abundant bacterial families. The columns on the left are the upstream water. The next column is the downstream water, that floating organic matter, and then the plastic is all the way on the right. And so if you sort of squint your eyes and look at this, you can see there's some significant differences in the patterns here representing differences in taxonomic composition. And there were a few things that stood out as being interesting. One is that bacteria from the family Pseudomonidaceae were really abundant. That's the red bar on the microplastic, much more so than the other habitats. Several members of the family Pseudomonaceae, they're common freshwater organisms, they're known biofilm formers, and they've also been associated with the breakdown of a variety of plastics. So they may be selectively colonizing the plastic in an attempt to break it down and use it as a substrate for growth. We also found high abundance of organisms associated with the human gastrointestinal system, including Veillonolaceae, which are not pathogenic but are common gastrointestinal organisms, as well as Campylobacteriaceae, which is a family that includes a number of gastrointestinal pathogens. So the presence of these gut-associated microbes on the plastic is not surprising because the plastic is coming through domestic wastewater, which is going to have a lot of human waste in it, going through the treatment system and ending up in the river. But this is a significant concern, right? Wastewater treatment plants are supposed to be removing gastrointestinal pathogens and human gut-associated microbes during the treatment process. So the concern here is the plastic providing a vehicle that allows these organisms to move through the treatment plant more effectively? And could they potentially be vectors to transport these pathogens downstream? So when we looked at that, compared the abundance of bacteria like Campylobacteriaceae, these potential gastrointestinal pathogens, they were most abundant immediately downstream, which is the downstream 50-meter plastic bar that's really high on that graph on the left. But even 2 kilometers downstream, the Campylobacteriaceae were still significantly more abundant than the other habitats. So these pathogens are persisting during transport through the river. And then the Pseudomonaceae, those biofilm formers that are associated with plastic breakdown, they're becoming even more abundant during transport through the environment. So this potential for the microplastic to be a vehicle for the movement of pathogenic bacteria is a significant cause of concern. Now, all the data that I'm showing you so far was just from one river and from one treatment plant. So in order to see if these trends were more generally observable, we studied 9 rivers in northern Illinois, each of which had their own treatment plant on them. And we saw that, yes, indeed, in most of the cases, the abundance of microplastic was significantly higher downstream, confirming that treatment plants more broadly can be point sources of microplastic. And when we analyzed the bacterial communities, we saw similar trends to what we'd seen in the single river study. All the microplastic communities are highlighted here in yellow. The floating organic matter or cestone samples are in blue. So you can see across all of these 9 rivers, the microbial communities colonizing the microplastic were different than those in the other habitats and were similar to each other. And when we looked at the individual taxa that were present, we saw similar trends to what we'd seen in the Chicago River, right? Where again, the Pseudomonas are really abundant on the microplastic. These are the potential microplastic or plastic degraders. And then the pathogenic Campylobacter were more abundant on the plastic, and the gastrointestinal associated organisms, the Veillonolaceae, were also more abundant. So those trends that we'd observed in the Chicago River were consistent across those 9 rivers that we studied in northern Illinois. So the take-home message from some of these early studies was that the wastewater treatment plants are point sources of microplastic to freshwaters, and that the microplastic represents a distinct microbial habitat that supports a microbiome that can include things like potential plastic degraders, as well as gut-associated and potentially pathogenic microbes. So these microplastics are coming through the domestic wastewater system and are going through wastewater treatment plants. So what we wanted to understand then is what's happening to the plastic as it goes through the treatment plant. Is all the plastic making its way through the plant, or is some of it being retained? And then what's happening to these microplastic associated microbiomes? The treatment plants are designed to try to remove these pathogenic organisms and the gut-associated microorganisms. Is the plastic creating the potential for these gut-associated and pathogenic microbes to make their way through the treatment system? So we analyzed samples from 2 suburban wastewater treatment plants in Illinois, and we collected sample from the raw wastewater, the raw sewage. We collected microplastic from that. We collected it from the final effluent going out to the rivers, and then we collected it from the sludge, which is the product that's produced from wastewater treatment. And when we analyzed the data, we saw that both of these treatment plants retained almost all of the plastic. So more than 99% of the plastic was retained by these wastewater treatment systems, which similar numbers have been shown in other studies as well. However, even with this high retention, because of the high volumes going through the treatment plants, there are still significant numbers of microplastics that are being released. And also, the other interesting point is that when we say the microplastic is being retained in the treatment plant, what that means is that it's being concentrated in the sludge. So the concentrations of microplastic in the sludge were incredibly, incredibly high. Now, keep in mind, in Illinois, most sewage sludge ends up eventually being land applied. So this could represent another potential route of entry for microplastic to the environment, something that should be potentially studied. When we analyze the microbial communities on the microplastic, what we could see is that they underwent a change, that the microbial communities on the sewage, which are represented by the circles in this graph, and the different open and closed shapes represent the two different treatment plants. The microbial communities on the sewage were different than the microbial communities that were released on the plastic in the effluent. So that small amount of plastic that made it to the effluent, the communities of microbes on there were different than what we saw in the sewage. And similarly, they were different in the sludge. So as the microplastic is moving through the treatment plant and being partitioned either to the effluent or to the sludge, the microbial communities are changing. And some of the changes that we noticed, which are highlighted in this graph, included, now this was interesting, the Campylobacter, which we'd noted on samples from the river before, was actually significantly reduced by movement through the treatment plant. So that's the red bars in this stacked bar graph. Campylobacter was really abundant on the raw sewage microplastic, but was significantly reduced during transport through the treatment system. So in this case, this pathogen is being reduced significantly by wastewater treatment. But another group of organisms, the Enterobacteriaceae, which is a family that includes many gut-associated microbes as well as a number of gastrointestinal pathogens. These are the light greenish bars on my stacked bar graph. They significantly increased in abundance during transport. So they were relatively low abundance on the plastic in the sewage, but then by the time they got out into the effluent, they were more abundant on the microplastic. So it seems like some pathogens are being reduced in abundance on the plastic by treatment, and some of them are actually being increased. And then Pseudomonas, that one that's potentially associated with plastic decomposition, also increased in abundance with transport. So, and these are just some of the numbers that back up what I just showed you. You can see the Enterobacteriaceae, again, increasing in abundance with transport. Archobacter, a genus from the Campylobacteriaceae family, decreased in abundance, and the Pseudomonas increased in abundance. So the conclusions are that wastewater treatment plants retain the vast majority of the microplastics that enter in the raw sewage, but the microplastics end up getting concentrated in the sludge. The transport of microplastics through the treatment plant alters the microbiome. Some pathogens are reduced in abundance, but some of them actually increase in abundance. So it is possible for the microplastic to still have significant concentrations of gastrointestinal pathogens once they've gone through treatment. So that's all I have for you today. I just want to thank my collaborators. I already mentioned Dr. Tim Holine. I also want to thank Dr. Sherry Mason, as well as our other collaborators, Amanda McCormick, Max London, and my friend John Scott, who I think is on the call today, or in the Zoom today. And then thank funding we got from Loyola University Chicago, the Illinois Water Resources Center, and the Illinois Indiana Sea Grant. That's it. Thank you very much.
We have time for one question.
Okay.
Great.
Very interesting that not all of the wastewater treatment plants had a higher concentration downstream. Were you able to make any conclusions as to why that was the case for certain wastewater treatment plants? Different treatment process, smaller facility, river flows, etc.
Yeah, I think the reason why we didn't see it for some of the sites, so when you start getting out into suburban areas in northern Illinois, many of these rivers have more than one treatment plant on them. The plants tend to be a little bit smaller. They're more like suburban treatment plants. So some of these rivers, our upstream sites, actually had treatment plants that were much farther upstream as well. So it's possible that they could be another source. There's also the potential for the runoff to be sources of microplastic or combined sewer overflows. So we're sort of oversimplifying by saying that in every one of these cases that the treatment plant effluent is the dominant point source. They all seem to potentially be point sources, but there are other potential sources as well. The one other point that was interesting is that in comparison to our two treatment plants, one of them used a sand filter for the effluent prior to release, and the other one didn't use a sand filter. And the sand filter did seem to enhance microplastic retention, that the retention was slightly higher at the one that had the sand filter versus the one that did not.